Bismillah ar-Rahman Rahim. We're going to start a discussion of a paper 3, which is the March 21, and we're only going to discuss question 1 in this video. So we start with the first question. Now, the first thing to understand is this is a 2-hour exam, and if there are 20 students taking the exam, half will have to do question 1 and half will have to do question 2 first, because you see, Cambridge only sends us half the number of slides for each group. So if there are 20 students, we will get 10 slides from Cambridge. So half of you will do question 2 and half will do question 1. So before you proceed carefully, read through the whole of question 1 and 2. So this is very important that you do read the whole question. I will of course not read it because I'm sort of solving it, but I would expect you to read the whole question. Please download it and read the whole question. Plan the use of the 2 hours to make sure that you finish the whole of question 1 and question 2. Agar cubes have been, agar is sort of a gel, and you actually get agar powder and we put it in water and it sets, it forms these, like you have jelly, you make jelly at home. So agar cubes that have been stained with universal indicator can be used to investigate diffusion. So understand that we're giving you this, we are investigating the process of diffusion, which is part of the chapter 4, which is cell membrane. When hydrochloric acid diffuses into the agar, so there is something, this agar block, which we have, and it's colored, we put it in the universal indicator, and then we put it in hydrochloric acid. So you'll put it in a beaker which contains HCl, and the agar cubes change color, and the end point is when it changes to pink. So basically it's going to change to pink. This is sort of a red but sort of a pinkish shade. Reach the whole cube has changed color to pink, the whole cube, and they have underlined that whole cube. I mean, it's in sort of dark letters. So whole cube, you will investigate the effect of concentration of hydrochloric acid. So now this, of course, when you change something, this is the independent variable. This is something you must learn to understand. What is the independent variable? What you change is the independent variable. I always say look at the word I, independent. And the other is called the dependent variable. This is always the result. The dependent variable is the result. And this is always plotted on the y-axis. So the mnemonic is dry. So dependent variable is the result and it's always plotted on the y-axis. So you are provided with the material shown in table 1.1. H is 1.0 mole dm cube hydrochloric acid. A is an agar block on a white tile. They've given it to you on a white tile. There's a reason for that, distilled water. Now, if H comes into contact with your skin, wash it off immediately. It is recommended that you wear suitable eye protection. Why? Because maybe if something by mistake, your hand, somebody passing by just nudges you and your hand sort of, the chemical sort of falls off and falls on yourself or in your face, but that could be disastrous. So eye protection is what you need to wear in the lab. That is something you must know. Then coming to the next part of the question, now it says you will use proportional dilution. Now there is something known as, you go onto YouTube and you can find these videos on simple dilution and serial dilution. There are lots of them. So you have to make a proportional dilution, which is called a simple dilution of 1 mole HCl. Now, of course, you have to realize that everything has to decrease after this. It can't increase from 1 mole. You can't make something 2 mole or 5 mole. You can't evaporate the water from it. So it's going to, the concentrations are going to decrease. To make the different concentration of HCl that you will need for this investigation, you will need to reduce the concentration by 0.2, and you will have to prepare 20 cm cube of each concentration. Now, using H and W. Now you can see here, I've done this for you. When it was 1 mole, 20 was H, and the water was 0. Now look, they're writing how they write 0.0, so you do it to one decimal figure. Now when it will be 0.8, you will do 16 and 4, 0.6, 12 and 0, 8 and 0, and 4, 8 and 12, and 0.2, 4 and 16. So what you have to understand is that how will you do this? I've shown you the maths working, which you're supposed to do is just a unitary method. 1 is 20, so 0.8 will be like this. And the total was 20, so the total must be, so 16 plus 4 is 20, 12 plus 8 is 20, 12 plus 8 is 20, and 16 plus 4 is 20. So this was 2 marks. This was something that means it's a very basic maths which every A-level student know. And if you don't know that, that's a very sad thing, because it's very basic maths. So then you were supposed to make these. So carry out step 1 to step 6. Prepare the concentration of HCl as shown. So you will have 5 beakers. Now you always make 5. When they ask you to make dilutions, please always remember to make 5. That's a standard rule which I ask you to follow. So you've got 5 here, 1.8, 1.0, 0.8. So you have 5 actually, 1, 2, 3, 4, and 5. So you have 5 concentrations. So you have 5 beakers and you label them 1%, and you label their 0.8 and so on and so forth, right? Prepare the concentration of HCl as shown. So you have 5 beakers, 5 concentrations, and you have 20 cm of each. So all of them have the same volume. All of them have the same volume. On the white tile provided, cut 3 agar cubes from the agar block. They've given you an agar block A for each of the concentration of HCl that you will use. Do not allow the agar block or agar to come in contact with your skin because this may affect your result. Then they've given you the measurement. They've probably given you a ruler as well. So you're now going to cut 15. Why have you to cut 15? Because you've got to put 3 in each. 3 cubes in this, 3 in this, 3 in this, and 3 in this. So you have to put 3 cubes in each. So you'll have to cut 15 cubes, and the size has to be 5 into 5 into 5 into 5. So you'll be given a ruler. Well, somebody will think about it. It's not going to be very exact. Some of you might do maybe a little 1 millimeter more or 1 millimeter less, because it's not very accurate. Ruler and with a sharp scalpel, it's not going to be a very easy thing to do. So you might, this is the ones which you're going to think of as the errors in the experiment. Then coming on to the next part, in step 3, you will put 3 of the agar cubes into the beaker. So here is the beaker, and you have to put the 3 agar cubes into it. And this is 20 centimeter cube. State the apparatus you will use to put the agar cubes into the beaker containing 1 mole. So it could be forceps. Don't say a spoon, because we don't give you a spoon. You could say a forceps. You could have said a spatula if it was given to you, but I think in the instruction it said that you have to give you a forceps. So you could have said a forceps, or you could have said a spatula. Then number 3, put 3 agar cubes into the beaker containing 1 mole and immediately start timing. Okay, so you put them in and you start the stopwatch, and you say, okay, fine, let's start the stopwatch. Measure the time taken for each of the 3 cubes to reach the end point. So maybe, I'm just cooking this up. So one took 10 seconds, then the other took 12 seconds, and the other took, say, 12 seconds again. So your stopwatch is going to stop at 34 seconds. And you will have mentally remembered it, that the first cube was 10 and the second cube was 12, and it was 12 again. So, what did you do? Did you do any other way? Did you stop the stopwatch and then start? But then you see, that means, would it stop at, what would be the total time on the stopwatch if it was 10, 12, 12? It would be 12 seconds. Because you didn't stop it at 10, so it's still running. And the second and third cube stopped it, so the reading on the stopwatch would be 12 seconds, not, as I said earlier, 34 seconds. I just want to see if you all were going to register that mistake. So you started the stopwatch at 10 seconds, 1 cube turned pink, at 12 the second and the third became pink. So 10, 12, 12, so the stopwatch is going to stop at 12 seconds. So put 3 agar cubes into the beaker, measure the time taken for each of the three to reach the end point, and record them in A3. The end point for each cube is when the whole agar cube has changed color to pink. Now this is again ambiguous. Pink is really pink, not pink, shade of pink. If any agar cube has not reached the end point after 3 minutes, then stop timing and record as more than 180. So that gives you a very good way of understanding that all the readings should be less than 180, because that was the maximum that you were supposed to wait. You can't say, okay, it took 10 minutes, because that's not allowed. You couldn't have said it's taken 5 minutes. because it says the maximum you were supposed to wait was 3 minutes, and then you had to just write in the record that it was more than 3 minutes. Then it says put three agar cubes into the beaker containing the next concentration. So in the next concentration, which was, if you did the first one was 1.0, then the next one was 0.8. So now you put the cubes that you had put there. Now you take fresh 3 cubes and you put them in 0.8, not the same ones. Of course, they've changed color. Then you put 3 agar cubes into the beaker containing the next concentration, and you prepared in step 1 and immediately start timing again. Then repeat step 4 and 5 until these results have been collected for all the concentrations. So that means all the 5, 1, 2, 3, 4, 5, 0.8, 0.6, 0.4, and 0.2. And you've got readings for all of these. Now, I am just assuming that the time increased. I'll tell you why the time increased and why I think the time should have increased. Because the most concentration was 1 mole. So in this, say it took 10 seconds. Now in 0.8, it probably took 15 seconds. In 0.6, maybe 20 seconds. So I'm increasing the time. 24 seconds, 36 seconds. So the thing which you've got to understand is that as the concentration was more, so there would be more molecules per unit volume and diffusion would be faster. So repeat step 4 and 5 in all the concentrations, and then of course comes here the important table which everybody must learn. Record your results in an appropriate table. Now if you look at this table, now this table was for 5 marks. Now it's very sad if anybody loses these 5 marks, because it's very simple. This, if you wrote concentration of HCl, and the units on the top, you got 1 mark. If you wrote time in seconds, you got the second mark. Then the third mark was for records times for each of the 3 cubes at each concentration. So giving these 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 12, 15. So 15 figures that I have written in yellow, you got 1 mark for that. And expected pattern of result was that, the HCl, the more the concentration, would probably be less time, and then it will increase with as the concentration became lesser as the diluted solution of HCl. And the result was given in whole seconds, not 10.2 seconds, not 11.4 seconds, not 13.6 seconds. Because your stopwatches have that provision, but we don't want you to write in fraction of a second. So if you wrote that in whole seconds, well, you got a mark for that. So 5 marks. I will be putting in the mark scheme at the end and going through it once again with you all. So this is the table, and this was the 5 marks. It says state the dependent variable in this investigation. Now, dependent variable was what were you collecting? What the results were you collecting? So that was time taken to change color. Time taken to change color. Or sometimes they can even ask you what is the apparatus used to find out the dependent variable. What is the apparatus used for you to record the dependent variable? No, that was a stopwatch. Then it says identify one source of error in step 2 to step 6. Well, of course, the error was difficult to judge the endpoint. It was very difficult to judge the endpoint of all the 3 cubes. Because you would put it in the beaker, the 3 cubes, and then you waited, and then you would say, okay, well, is this happening? It's taking 10 minutes, 10 seconds, and 11, then 12. And then you were also thinking of the first one, then said, okay, the first one's done, okay, the second one's done, okay, the third one's done. Well, you were a little confused. So you could have said the improvement could have been that use 3 people, 3 observers. Use 3 observers to identify the color change. So you could have said that. You could have said something else also. is record a video. If you recorded a video of it, that would exactly tell you how many seconds it took for it to change the color. And what else could you have said? You could have said, do it 3 times. I mean, don't put 3 cubes. But then, of course, the 3 cubes was part of the experiment, so you couldn't really change it that much. But another error could also have been in measuring the cubes. And for that, you needed to have either a vernier caliper gauge or something more exact to be able to measure it exactly that was 5 millimeter by 5 millimeter. So that is also an error, is the size of the cubes. Then coming to the next part of the question, it says a student suggested a hypothesis. What is a hypothesis? You must know what is a hypothesis. Hypothesis is a theory. The theory was, as the concentration of HCl decreases, the time taken for acid to diffuse into the agar will decrease. As the concentration decreases from 1 to 0.8, the time taken will also decrease. Now, either you support it or you don't support it. Now, I don't support it because my results are given in such a way that as the concentration decreased, the time taken increased. And how would you, so then I would give you the figures. At 1.0 mole and at 0.2 mole, this was say 12 seconds. And then it was say 40 seconds, whatever the result is. Now, this was the evidence that I needed. Only then I'm going to give you a mark for that. So it just does not support is not the mark. The mark is what is your evidence. That as in 1 mole it has taken 12 seconds, in 0.2 mole it has taken 40 seconds. So then of course this is against this hypothesis. In part 7, the procedure described in step 1 to step 6 investigated the effect of changing the concentration of HCl on the rate of diffusion, using the terms taken to reach the end point, using the time taken to reach the end point. Describe how you'd modify the procedure to investigate the effect of changing the temperature on the rate of diffusion using the time taken to reach the endpoint. Same experiment, but how would you change it for the temperature? So you would use the same concentration of HCl, and that I would like you to specify 1 mole dm cube. And so you used only one concentration, and you at least used 5 temperatures. But then writing this will not get you the mark. You'd have to specify the 5 temperatures. And you have to write the units as well. So 30, 35, 40 degrees Celsius and 45 degrees Celsius. So any 5, I'm not saying these, you could have written 30, 40, 50, 60, 70, you could have written anything else. And of course, how would you maintain the temperature? So you use a thermostatically controlled water bath. This is a very favorite sentence which I want you all to remember. Use a thermostatically controlled water bath. So this is a sentence which everybody must remember, thermostatically controlled water bath. B part of the question says salt is used to preserve a cheese. Cheese can be soaked in a salt solution to allow the salt to diffuse into it. A scientist soaked cheese in a 15% salt solution at 4 degrees Celsius and measured the distance that the salt had diffused into the cheese at intervals over a period of 20 hours. So this always, the first column is the independent variable, and it is always plotted on the x-axis. The next, which I always tell you to remember, is the second or the last column could be, this is the dependent variable. And this is usually the result that we have calculated, and it is always plotted on the y-axis. So this is a rule. First column, independent variable x-axis. Second column or the last column, sometimes there are a number of figures and the last column has to be plotted. The y-axis will always have the dependent variable. So this is what you have to do. So you have to plot a graph of this. Then we look at the grid given to us. So plot a graph. So plot a graph of the table 1.3 on the grid. Use a sharp pencil for drawing graphs. Now you have to distribute it on this. So first of all, what you do is you label the axis. So the axis labeling was time in hours goes on the x-axis. So you just wrote this. you just wrote time slash hours, whatever is written in the previous table, time hours. So use exactly what was written, no abbreviation. And then you wrote distance slash centimeter. So this would come on the y-axis. So I must get this right because you might think it's a ECM, right? Then you decide how much are you going to, what is the scale that you're going to take. The scale is the most important thing, and you need to label every 2 centimeter. So 5 to 2 centimeters. So on the x-axis, what was allowed by Cambridge was that this would be 5, this would be 10, 15, 20, and 25. This was the scale which Cambridge expected you to keep, because you have to remember on the x-axis we have 0 and 20. So 0 and 20, you have to spread the graph between 0 and 20. So 0 is here, and then on the y-axis, then again you wrote 0, and the y-axis scale was 0.2 to 2 centimeters. So this was 0.2, this was 0.4, this was 0.6, this was 0.8, this was 1.0, and then this was of course 1.2. And you have to label it every 2 centimeters. So this is 2 centimeter, 2 centimeter. You have to label it. I'm not saying put these lines, but you have to label the axis. Then you plotted the points. Now the points, which of course I'm just plotting it randomly, you had to either do a circle, put a dot on the circle. I've not exactly plotted it correctly. I'm not doing that. Or you could have put a cross. So either a dot and a circle or a cross. Now, whatever you decide to do, that is up to you. I'm not saying you do any one method is allowed. Both can be allowed, but don't mix them. Don't do a dot sometimes and then a cross sometimes. So, and then of course, somewhere here, I'm not doing the correct plotting. And then you have to join the point. So the best way is with the ruler join point to point. But you can do a curve as well. Both are allowed. Either you do it with a ruler, plot to plot with straight lines, or connected with a curved line. It says use your graph in figure 1.2 to calculate the mean rate of diffusion of salt during the first 3 hours. So this is what you have to understand was, so you would then, do a calculation on the 3 hours. You would draw a line and you would see where is the y-axis, and you would read off this. And this value, then you would divide this value by 3, because look what the answer is in. The answer is in centimeter cube per hour. Calculate the mean rate of diffusion during the first 3 hours. So whatever your value comes on the y-axis, you divide it by 3, because here you have to do a calculation centimeter per hour. So this is what you had to do for this part of the question. The scientists repeated the investigation at 15, found the rate of diffusion at 15 to be greater than the rate of diffusion at 4. Explain the difference. When it's explained, then it's biology. So it explained the difference. So it was a very simple thing. At 15 degrees Celsius, naturally, there will be more kinetic energy. So more kinetic energy will result in more collisions. And so molecules move faster. So more collisions. So the rate of diffusion will be faster. The rate of diffusion would be faster because more the temperature, more the kinetic energy, so the rate of diffusion would be faster. Now this is the thing which we do in O levels. We tell you energy needed for active transport, but energy not needed for diffusion. But then we say, okay, temperature increases the rate of diffusion. So there's something wrong. Actually, for ATP, we need ATP. We don't need energy. We can't increase the temperature and increase the rate of ATP, rate of active transport. You see, active transport requires ATP. Active transport requires ATP. But energy for diffusion is not such as such required, but molecules need energy, the kinetic energy to move. So the rate of diffusion will increase if the temperature increases. So this is a very, very interesting thing which you need to now sort of understand at A levels. Active transport requires ATP. In O levels, we say active transport need energy. Well, that's wrong in a way. Now we don't say that anymore in A levels. So diffusion needs energy. Yes, because increasing temperature increases the rate of diffusion. And diffusion is the random movement of molecules and they move from a high concentration to a low concentration. Okay, that finishes this question which we discussed in a previous class. And so now I'm putting up this video for all of you to understand and comprehend. And thank you very much.